natin. Pwede na siguro yan. Let's go on. So we're going to discuss the ETS, the electron transport chain. Na-upload ko naman yung PowerPoint, tapos may extrayer naman kayo. So, madali na lang naman sundan to. I'm going to share my PowerPoint. Okay, so this is the electron transport chain. So this the material here came from extrayer. So basically, di ba kung natatandaan niyo pa, from the Krebs cycle, you have generated lots of NADH and FADH2. So basically, sa electron transport chain, dito nako-convert yung NADH at saka FADH2 sa ATP. Basically, ganun lang naman. Kasi ang explanation niyan, yung kasing mga electrons sa NADH2 at saka sa FADH2 ay high potential. They form a band. So when you cut this band, it will release lots of energy. Parang ganun yung explanation. So this energy is catched by the ADP and the ADP is converted to ATP. Parang ganun lang naman yung kwento ng electron transport chain. So in the process, in this biochemical pathway, you're going to produce ATPs and yung electron will be transferred to oxygen and then your oxygen will be converted to water. So kung yung carbon dioxide galing sa Krebs cycle, yun namang water, yun namang water ay galing sa electron transport chain. Kasi 'di ba when you oxidize, when you oxidize the fuel, the glucose, it will become carbon dioxide and water. Pag nagkaroon ng respiration. So the final product is carbon dioxide and water. So let's take a look at the detail. Electron transport chain. So the other name for electron transport chain is oxidative phosphorylation. So this is the process in which ATP is formed as a result of the, dali lang baka marami na sa aming open track. Okay. So oxidative phosphorylation is the other name for electron transport chain. So, kasi oxidation siya. Tapos phosphorylation. Phosphorylation refers to the conversion of ADP to ATP. So oxidative phosphorylation is the process in which ATP is formed as a result of transfer of electrons from NADH or FADH2 to oxygen by a series of electron carriers. So yung mga electron carriers, yung mga enzymes na di-discuss natin. Okay. So oxidative phosphorylation generates 26 of the 30 molecules of ATP. So dito nang gagaling yung 26 out of the 30 molecules of ATP that are formed when glucose is completely oxidized to carbon dioxide and water. So siyempre, if you're going to trace, saan ba nanggaling yung apat pa? So yung dalawa galing glycolysis, yung dalawa pa, yung GTP, na actually nako-convert din sa ATP sa Krebs cycle. Pero yung 26, galing yun sa sa electron transport chain. So for every NADH, it will become 1 NADH will become 2.5 ATP. Tapos 1 FADH2 will become 1.5. Depende sa libro, pwedeng 1.5 ATP. So kung natatandaan niyo yung Krebs cycle, basically you're going to produce 4 NADH kasama yung pyruvate, to acetyl-CoA, di ba? Tandaan niyo pa 'yon. So you have 4 NADH sa Krebs cycle. Tapos times 2 kasi 'di ba dalawang pyruvates, So all in all 8. So 8 times 2.5 ilan 'yon? Mga 20, So you have 20 sa NADH pa lang. Tapos yung FADH2, how many FADH2? Bale, isa so dalawa. So that's 3. So that's 3 tapos probably in this particular conversion paano ba? Kulang pa So 3 FADH2. So 1.5, 1.5, that's 3. Meron pang tatlo. So probably I'm missing one NADH. Ano, parang may NADH ata sa glycolysis. Ano? So doon galing yan. May NADH na napoform sa glycolysis. So paki-check nyo na lang. Parang may NADH pa na isang napoform sa glycolysis. So doon galing yung ano, yung 3 ATPs. We're going to check that later. So anyway, basta what is important here is generally most of the ATP is formed in the electron transport chain. Ano? May ano rin kasi yan eh, depende sa reference, magkakaiba din yung conversion ng number ng ATP from NADH at saka from FADH2. Meron pa rin ganung issue. Ano? Kuminsan pati yung 30 molecules na yan, kuminsan yung ibang libro ang count nila dyan 32. So, ano yan? Ano nagbabago-bago depende sa reference. Okay, so oxidative phosphorylation is conceptually simple, no? Kasi basically ang nangyayari lang naman dito, high potential yung electron. So because of the high potential of electron, you can produce lots of ATP. Parang ganun lang naman yan, ano? Parang you're just going, parang kung halimbawa sa ano sa tubig, yung tubig nyo nasa dam, mataas yung kanyang potential energy, tapos pinapabayaan nyo lang siyang bumaba sa dam, so you're going to produce electricity. Parang ganun lamang yung concept ng why oxidative phosphorylation is conceptually simple. Pero sabi nga dito, it's mechanistically complex kasi you will see it involves lots of molecules, big molecules, enzymes, para ma-accomplish yon. Tapos dahan-dahan. So because of that dahan-dahan na pagta-transfer ng electron, dahan-dahan siyang bumababa, it becomes so efficient, no? So actually hanggang ngayon hindi pa natin kayang ma-achieve, no? Ma-achieve ng human using our artificial machines yung efficiency ng ng biological world. Mas efficient to, no, compared sa pinag-aaralan natin sa thermodynamics, ano? So mas efficient pa rin yung biological world. Okay, so yun yung ibig sabihin ng phrase na oxidative phosphorylation is conceptually simple but mechanically complex. You will see the mechanical complexity of ETS later. So sabi dito ni Stryer, the unraveling of the mechanism of oxidative phosphorylation has been one of the most challenging problems of biochemistry. No? So parang 1960s na, hindi pa nila talaga ma-discover kung paano nangyayari yung electron transport chain. So makikita nyo later, merong si Peter Michel, he discovered the ATP synthase, kung paano nangyayari yung process using the chemi-osmotic theory, no? So nanalo siya ng Nobel Prize ng 1970s at saka pa lang na-elucidate yung electron transport chain, no? So ganun kabigat ng Nobel Prize sa chemistry. So oxidative phosphorylation in eukaryotic takes place in the mitochondria. So dalawa kasi yung cells, merong eukaryotes at saka yung may prokaryotes, kung natatandaan nyo pa ng high school. Yung eukaryotic cell, ito yung merong mga mitochondria, yung may mga organelles. Tapos yung mga primitive cells like yung mga prokaryotes, wala yung mga mitochondria. So yung mga prokaryotic cell, hindi sila nag-undergo ng oxidative phosphorylation. Glycolysis lang. So pag may mitochondria, the oxidative phosphorylation takes place in the mitochondria. So kung natatandaan nyo pa yung mitochondria, di ba may dalawa siyang membrane, the outer membrane and the inner membrane. Actually yung mga enzyme ng ETS nandoon sa inner membrane, naka-embed doon. So Eugene Kennedy and Alfred Leninger discovered a half century ago that mitochondria contain the respiratory assembly, no? The enzymes of the citric acid cycle and the enzyme of the fatty acid oxidation. Okay, si Leninger yung author ng biochemistry book. Nandito daw sa loob ng di ba nasa matrix na pag-aralan natin nung discuss natin yung Krebs cycle. Nasa matrix yung enzyme ng Krebs cycle. And then later makikita niyo pag nag-discuss tayo ng beta oxidation, ando din yun nasa matrix. So unlike sa oxidative phosphorylation, nasa ano yan, nandito siya sa inner membrane, nasa inner membrane yung mga enzyme ng oxidative phosphorylation. So oxidative phosphorylation takes place in the inner mitochondrial membrane, in contrast with most of the reaction in the citric acid cycle and the fatty acid oxidation, which takes place in the matrix. No? So makikita nyo mamaya kung bakit kailangan nandoon sa matrix. Kasi ginagamit niya yan, ginagamit niya tong matrix at saka yung intermembrane space. Doon nangyayari yung chemiosmotic process. So, paano ba nagkaroon, ito yung parang evolution theory, paano nagkaroon ng mitochondria yung mga prokaryotes? Paano sila nagkaroon ng organelles? So, ang theory ng mga evolutionists, dati daw yung mitochondria, they are independent organism. Tapos in-engulf lang sila ng mga much bigger pre-living organism. Tapos naging part na sila nun, no? So, a pre-living organism capable of oxidative phosphorylation was engulfed by another cell. So yung proof nila, parang yung composition, yung composition ng mitochondria is very similar dun sa nag-i-exist na mga prokaryotic cell like Rickettsia, Prowaziki, yung nagkakos ng typhus. Mukha siyang mitochondria. Okay? Ito yung theory nila kung paano nagkaroon ng mitochondria. It's dati daw sa kwento parang dati they are independent microorganism na na-engulf ng mas malalaking microorganism. And then yung proof, yung genetic composition ng mga existing microorganism na maliliit. Okay. So, why is this possible? So, this is possible. How do you convert why is it possible to convert NADH to 2.5 ATP? So as we go along, we're going to take a look at the thermodynamic potential, no? So for example, yung conversion ng NAD+ to NADH, of course it involves electron, di ba? Kasi magiging NADH yan. So yung conversion nito, ang potential niya is yung voltage niya is negative 0.32 volts. Of course, alam niyo naman na NADH is energy filled. So yung forward reaction na to is non-spontaneous, kaya nga negative yung kanyang voltahe. Pero yung reverse, positive, no? It's spontaneous. So yung conversion ng NADH to NAD+ is spontaneous. Actually, yung kanyang potential is negative 0.32. And then, of course, I hope you still remember this formula ng delta G. So delta G is equals to N is the number of electron, F here is your Faraday's constant. Is to remember Faraday's constant yung 96,500. Tapos ito yung delta E. Ito yung negative 0.32. So pag kinumpute nyo yung potential, no? Yung potential ng remember yung potential ng ng NADH becoming NAD+. So palitan nyo lamang yung F ng 96,500. And then as you can see, we're using 2 electrons. So yung N dito is 2. So negative, just grab my calculator, negative 2 times 96,500. So tandaan nyo pa naman, this is voltage per coulomb, yung F natin. Coulomb pala, coulomb per voltage, sorry. Coulomb per voltage. So pag multiply nyo ng times 0.32, positive na yan kasi yung baliktad yung kukunin natin. So times 0.32, you'll get the this one, the 61,760. Ito yon. So divided by 1,000, you're going to get the 61.7 kilojoules per mole. Dapat may negative to. Kung natatandaan nyo pa, may typo error lang. May negative to dapat. Pag ang delta G ay negative, spontaneous yung reaction. So yung reverse nito is spontaneous. Ito yung kanyang potential. Negative 61.7 kilojoules per mole. Hindi pare-parehas tayo, hindi tayo familiar dito sa 23.06. This is coming from this is coming from Stryer. Kayang-kaya niyo to. Yan? Biochemistry book. You are a chemical engineer. Kayang-kaya niyo. Any question regarding the calculation of delta G ng NADH? Okay, let's compare that dun sa ATP later, So aside from that, ang mangyayari pa yung oxygen will be converted to water. And then if you're going to check the potential table, yung potential nito is 0.82 volts. No? So pag inad nyo pa yung yung positive 32 ng NADH, you will have a 1.14 volt potential. So overall, ito yung potential na nagda-drive sa conversion ng NADH to ATP. Kaya nga sinasabi ni Stryer, ETS it's quite simple. Yung kanyang is conceptually simple. Kasi kitang-kita mo agad yung taas ng potential, no? Nung process wherein you just convert NADH and oxygen to ATP. Ganun kataas, no? 1.14 volts. Diba parang meron kayong battery. Actually parang meron kayong battery, ano? So pag kinonvert niyo yung dalawa, yung sa oxygen at saka yung sa NADH, you're supposed to get negative 220.1. Tama? Yung 1.14, tapos i-multiply niyo siya ng 96,500, tapos dalawang electrons din yung involved. Okay? Pati sa oxygen, di ba, dalawang electrons din yun. Ito, dalawa din to, ano? So constant lang yung ano. So if you're going to do that mathematically, you're going to get the 220,020. Nakuha niyo to, yung negative 220,020. And then compare that dun sa potential ng ATP. Parang ADP, are you following? ADP to ATP, you only need 31.4 according to Stryer, you only need 31.4 kilojoules per mole. This is hydrolysis of ATP. Ibig sabihin, ATP to ADP. So pag binaliktad nyo to, positive to. Ano? Okay? So actually, pag tinignan nyo, sobra-sobra pa. Kasi ang assumption natin 2.5 lang. Ano? Ito nga actually 31 lang. So 31 times, times 3 na lang. Wala pang 100. So sobra-sobra talaga yung potential, no? ng NADH at saka oxygen para makapag-produce ng 2.5 ATP. Ganun lamang yung simple thermodynamic analysis in terms of delta G. No? Can you follow? Do you have any question at this point? Malinaw naman. Hindi naman kayo nalilito bakit hydrolysis of ATP negative. Kasi ibig sabihin nito ATP to ADP, kaya negative yun. Spontaneous yun. Pero pag binaliktad nyo, kasi we're going to produce ATP, pag binaliktad nyo, magiging positive to. Ito talagang negative to, tama to. Ano? Kasi this is NADH and oxygen to NAD plus and water. Okay lang? Questions? Tanong lang kayo. Okay. So, sige, daanan na rin natin to. So, this is the calculation formula yung makatulong sa board exam nyo, no? This is the calculation for, halimbawa, meron kayong pH system. Tapos you wanted to, meron kayong galvanometer, meron kayong pang-measure ng voltage. Okay? So, this is the formula in calculating the pH, no? Tapos given yung meron kayong galvanometer lang yung gamit nyo, hindi talaga pH meter. So this problem comes from analytical chemistry book. Pangalan nito. Basta medyo nakalimutan ko yung pangalan ng analytical chemistry book. SDen. Tandaan niyo pa? Analytical chem? Anyone? Anyway, the cell, so this is the SCE, SCE has a potential of 0.2106 volts. When the solution in the right-hand compartment is a buffer of 4.006 volt. The pH is 4.006. So ito yun, ito yung pH na yun. Tapos yung kanyang potential, ito yung E. So with this first pair of data na given, ika-calculate nyo lamang yung E na. No? So just use your calculator tapos shift solve. Halimbawa, 0.2106 tapos alpha equal tapos alpha x tapos minus 0.0592 times 4.006, no, tapos shift solve. You're supposed to get 0.4477. Nakuha niyo ba? 0.4477. So that's the E naught. So tapos the following potential are obtained when the power is replaced with unknowns. So halimbawa, if the current voltage reading is negative 0.2902 volts, ano yung pH nung solution na pinalit nyo? Parang ganun yung question. So, yung nakuha yung E naught, gagamitin nyo na ngayon yun. Tapos ito, itong negative 0.2902, i-substitute doon sa E. Tapos yun, you're going to solve for the pH. Anong pH? Sige nga, try to work on it. So, pasok niyo lamang yung negative 0.2902. Tapos alpha equal, yung answer kanina, kung ano man yung inat nyo. Tapos minus 0.0592. Okay? Tapos times x. X yung pH nyo ngayon. Anong lumalabas? What's the pH? Jenilyn.
Magluluto ko na Mars and
What's the pH? Anyone? Sino ba? Michelle, anong pH natin? Na-encounter niyo ba itong problem na to? Rosel, Rosel, Miss Morpe, ka-approve ko lang thesis mo. Ano pH yan? Miss Rosel, anong pH natin? Trisha, ito si Trisha mabait. Anong pH natin? Ibita, anong pH natin? Yun. Trisha, 12. Tama, 12. That's 12.47. Ano? So, ganun lang, ano? So, gagamitin kasi natin sa susunod na slide. With this equation, ano? So, kasi aside from the conversion of ADP to ATP, meron kasing nangyayaring pumping of H+, no? So aside from kaya meron pang ibang ginagamitan ng energy. Yung kasing H+ nyo, ito transfer nyo from the matrix of the mitochondria papunta dun sa intermembrane space. Na-imagine nyo ba? Di ba, we have two membranes sa mitochondria. So, ang generally situation kasi, maraming H+ doon sa intermembrane space. So, yung transfer ng H+ from the matrix to the intermembrane space will need some energy, no? So, kaya it consumes energy. So, gagamitin natin yung calculation natin kanina, 'di ba, yung ginagamit sa pH meter in order to calculate the potential, no? So it's given from Stryer that the pH is 1.4 units lower than inside. So itong 1.4 units i-convert natin sa volts, no? So yung kanina, 'di ba, E is equals to E naught tapos minus 0.0592 times the pH. So yung 0.0592, i-multiply niyo sa 1.4, 1.4 units. Lag pa pala, lag pa. pH is 1.4. 0.0592 times 1.4. Tama, this is kilocalorie. Tapos yung 0.14 volts. So, I'm expecting na pag minultiply ko yung 0.0592. Well, anyway, basta yung 0.14 volts, yung 0.14 volts, the membrane potential is 0.14 volts, the outside being positive, ano? So, yun times 1 times 96,500. So this is 13,510, ano? Tapos this is in joules. So pag dinivide ng 4.184, 3.2 kilocalorie. Meron pa talagang difference. So balikan na lang natin siguro pa next meeting natin. Okay, so let's go to the enzymes, ano? So ito yung kwento ng enzyme. So the respiratory chain consists of 4 complexes, ano? 3 proton pumps and a physical link to citric acid cycle, ano? So kung natatandaan nyo, kung nung high school meron tayong complex number 1, complex number 2, complex number 3, at complex number 4, and then yung ATP synthase kung minsan tawag nila din complex number 5, no? So basically yung NADH, ang mangyayari dito, may electron to na high potential, ipapasa niyo yun sa Q, sa ubiquinon, no? Tapos yung enzyme na tumutulong sa pagpapasa ng electron, we call that the NADH Q oxidoreductase. Yung NADH Q oxidoreductase is also known as the complex number 1. Tandaan niyo pa? Na-discuss ba to sa ano? Sa high school ano niyo? Chemistry? Kasi natatandaan ko, tinatanong na to sa mga quiz contest sa science quiz contest sa high school. So, may possibility na discuss na to sa inyo. Ano? So, bakit ganito yung pangalan ng enzyme? NADH Q oxidoreductase. Kasi yung, di ba tandaan nyo pa yung Geroa, Leora? So, Geroa, when you gain electron, Geroa, R. The species is reduced, and then that's the oxidizing agent. Tandaan niyo pa yung Geroa, Leora in your chemistry. Pag Leora naman, when it lost electron, it is oxidized. So NADH will lose electron, no? So that's why oxido. NADH is oxidized. Ganun yun. NADH oxido. And then yung electron ng NADH pinapasa niya sa ubiquinon, sa Q. So that's why Q is reduced. So that's why the name of complex number 1 is NADH-Q oxidoreductase. Ganun yun. Ano? Tapos ito namang succinate Q reductase. Ito naman yung ginagamit ng FADH. No? Ito NADH. Dito naman sa succinate Q reductase, ito yung complex number 2. This is complex number 2. Dito naman yung FADH2. So FADH2, tapos tinatransfer niya sa ubiquinon. So Q is reduced kasi nalagyan siya ng electron, no? Kaya yung pangalan ng complex number 2 is succinate Q reductase. This is the same enzyme na nagko-convert sa succinate to succinyl-CoA sa Krebs cycle, ano? Kasi ito na yung ubiquinon. So yung ubiquinon, that's why the complex number 3 enzyme is named as Q cytochrome C kasi yung electron ng Q, ipapasa niya sa cytochrome C. Cytochrome C is another protein complex. Puro protein complex yan. Ano? So kaya ulit yung pangalan niya oxidoreductase. Kasi Q or ubiquinone is oxidized. Tapos yung cytochrome C naman gains electron. Kaya merong cytochrome C reductase. No? So Q cytochrome C reductase. This is complex number 3. And then the cytochrome C will be oxidized. So that's why we have this complex number 4. So yung complex number 4 natin, cytochrome C oxidase. Kasi natanggalan ng electron yung cytochrome C, pinapasa niya sa oxygen. And then oxygen, because of this extra electron, will become water. So yun, makukumpleto na yung product ng oxidation natin. Ano? Water at saka carbon dioxide coming from Krebs cycle. So parang ganun yung istorya ng ng ATS cycle. Let's take a look at the detail. Pag hindi natin natapos, tuloy na lang natin next meeting. Sa Monday, ano? So the high potential electrons of NADH enter the respiratory chain at NADH-Q oxidoreductase. Tandaan niyo pa yung Krebs cycle, nag-produce ng NADH. So papasok sa ATS, tapos there's the enzyme, no? Nandito naka-embed dun sa inner membrane. Remember the inner membrane of the mitochondria? So this is also known as complex 1 or NADH-Q oxidoreductase. Question? So this is your ubiquinon, no? So nandoon din yan sa sa intermembrane state. Nandoon din sa intermembrane, nakakabit din siya dun sa dun sa inner membrane ng mitochondria. So ito yung matrix sa baba, yung sa taas yung intermembrane space. Ano? So this is your ubiquinon. Okay, so this is the process. It absorbs electron, tapos nagbabago yung structure ng ubiquinon. Ano? Pero bago dumiretso yung electron sa ubiquinon, una muna pumupunta siya sa plavins. These are your plavins. No? These are your plavins. Remember the riboflavin, vitamin B2. No? So these are your plavins. Dito muna dumadaan yung electrons sa plavins. Tapos dadaan pa sila sa iron sulfur cluster. Tapos tsaka pa sila dediretso sa ubiquinon. No? Ganun yung flow ng electron. So that's why it's mechanically complex. Ano? So bago na siya ma-transfer sa Q, sa ubiquinon, meron pang dadaanan na plavins at saka iron sulfur complex. So ito yung general reaction sa complex number 1. You have 1 mole of NADH, tapos 1 mole of ubiquinon. Aside from being aside from facilitating the transfer of electron, yung isa pang ginagawa ng complex number 1, it pumps, no? It pumps H+ from the matrix papunta dun sa inner membrane space, cytosol, ano? Cytosol yung other term dun sa inner membrane space. Remember that that's the space between the outer membrane of the mitochondria and the inner membrane of the mitochondria. Yung inner membrane, ano? Okay? So you're going to produce 1 NAD plus, and this is the reduced ubiquinon, yung QH2. Ano? So, yun, nagpa-pump din siya ng H+. And then later yung pinapump na H+ na yan, yung dadaan sa dadaan sa ATP synthase. So because of that, kasi positive charge yun, ano? Because of that positive charge, yun yung nagda-drive para yung ADP maging ATP. No? So later makikita niyo yung complex number 5. So complex number 1 pa lang tayo. So pero basically ito yung importante dito, ano, yung pinapump na H pluses from one place to another. Yan yung nagki-create ng potential. 'Di ba parang when electricity lang naman kasi, 'di ba, flow of charges lang naman yan. So when a process results to a flow of charge, nagpo-produce siya ng energy. Parang ganoon lang naman basically yung sinasabi, ano? Okay, so ubiquinon Q is the entry point for electrons from FADH2. So yung, 'di ba, meron tayong FADH2 from Krebs cycle. So papasok 'yon sa ATS using complex number 2. Complex number 2 is succinate Q reductase. That's the other name, ano? So ganun din, yung electron ng FADH2 mapapasa din sa Q. Actually the same story. Hindi diretso sa Q, dadaan pa rin sa plavins, tapos iron sulfur complex, tapos tsaka pa lang diretso sa ubiquinon. And then the ubiquinon, pag meron na siyang H2, ang tawag na sa kanya ay ubiquinol. Walang ayupak sa biochem. Hanggang ngayon, common name pa rin ang ginagamit. Hindi successful yung adoption ng ayupak sa hanggang to the point na inaccept na rin ng ayupak yung mga common name ng biochem. No? Hindi nila talaga. Kasi mas komplikado. Okay, so complex number 1, complex number 2. Nasa complex number 3 na tayo. So sa complex number 3, kung na-imagine nyo, yung electron nasa ubiquinon na, nasa Q na. So ang gagawin ng Q, ipapasa niya yung electron sa cytochrome C. Sabi niyo, bakit ba pasa-pasa? No, parang basketball. Kasi mas mas efficient pag dahan-dahan. Pag binigla mo kasi, hindi siya efficient. No? Alam niyo naman yan sa thermodynamics, 'di ba? Kung gusto mo reversible, dapat dahan-dahan, konti-konti. Tatandaan niyo pa yung mga reversible process, 'di ba? Maliit lang yung diferensya. So ganoon din yung in-apply ng biological system, dahan-dahan. So ang ginawa nga lang ng mga chemists, ng mga biochemists, pinangalanan nila bawat protein complex. So that's why madami lang pangalan, pero basically, yun, pababa yung potential ng electron. No? So from Q to cytochrome C, so yung pangalan ng complex number 3 is Q cytochrome C oxidoreductase. Or pwede ring shortcut cytochrome reductase. Kasi yung cytochrome nagkaroon ng electron, so that's why it's cytochrome reductase. I hope, alam niyo naman yung Geroa, Liora, ha? Hindi tayo nagkakalituan Yan yung pinakamadalas na lumalabas sa sa board exam. Lilituhin kayo sa Geroa-Leora, oxidizing agent, reducing agent, punong-puno ng ganyan. Pag hindi niyo yan naintindihan, hindi kayo papasa sa board. Nung ako nag-take ng board, master na master ko yan, Geroa-Leora lang. Kaya, ako madalian lang ako sa board. Okay, so these are the active parts of the of the complex number 3. We have the rescue iron sulfur center, the Hemi C1, the Hemi BL, and the Hemi BH. Basically, itong mga parts na to, ito yung gagamitin doon sa explanation kung paano nagwo-work yung enzyme. Rescue, Hemi C1, Hemi BL, Hemi BH. So, paano ba nata-transfer